Well, thank you for having me. It's a delight to be here and hopefully help answer this question. So the answer is, spoil it, yes, no, or maybe, depending on your family history needs and kind of what's available. So we'll go through and talk about some of those things today. So I thought I'd begin first with just a brief introduction of NIST. If you weren't aware of, that's the place off exit 10 off 270. That's been since 1901, the National Bureau of Standards, and we changed the name to National Institute of Standards and Technology in 1988 when computers, requirements to help with computers were added to that. And then our main campus is what's in Gaithersburg, just a few miles from here, and then we have a big campus in Boulder, Colorado. That's where the atomic clocks are that define time.gov and the official time and everything. So we have many reference materials. One of the ones that I've worked on is a DNA reference material to make sure that DNA comparisons are done across forensic laboratories and clinical testing laboratories in a consistent manner. So this particular standard allows laboratories to calibrate their results. Just a DNA sample that's been well studied. So I think one of the things that you hear a lot in the news about DNA, we'll talk about some of the more recent news with genealogy and forensic connections that are starting to be made, but I like to start this. This is John Grisham's novel. Has anybody read this, The Whistler? I don't know if anybody likes Grisham, but he has a lot of interesting things he puts in his books. And this one, he has a single source blood stain that's collected from a crime scene. He has the blood stain being generated from the DNA being generated from the blood stain in less than 2 hours with rapid DNA testing. And then he has a search of the state database in less than 30 minutes. Links the crime profile to a previous offender whose DNA was in the database and helps solve the crime. And then this information is used as intelligence to help stop a corrupt judge and take this person off the bench. So the question...
You ruined the ending.
Yes, well, sorry. So certainly the first one is possible. They've been getting DNA from bloodstains for forensic testing since 1985. The second one, the rapid DNA testing, is certainly possible as well. There's a lot of technology development in the last 10 years that's enabled this. And 2 years ago, we've had a law put in place now called the Rapid DNA Testing Law that allows DNA testing now to be done in booking stations. So that's something that's starting to roll out and will be growing in the coming years. And then searching the state database, we've had a national database and databases on state levels for the last 20 years. And so that certainly can be done on a very rapid level. And the last one, well, that depends on the book, I guess, and the story. So my background, as mentioned, I worked at the FBI for a few years, about 25 years ago. I worked for a startup company in California, and then I came to NIST, first as a postdoc, and then after the time in California, to come into back here to Gaithersburg. I love teaching, and those are my 5 books on DNA. And then I also have a website called Starbase that helps you learn more about short tandem repeats, which are the markers that are being used in both forensic testing and some genealogical testing. We'll talk about that a little bit. And then I've helped with training. This is a material I prepared about 2 years ago to help with training judges in England on how to understand DNA testing. So I was one of the writers for that. So anyway, so family history is about families, and so this is my family a couple years ago, and we went to, my parents live out west on a ranch, and this is a gathering of them, and my siblings. So they've even numbered their posterity. 10 years ago we had a family reunion here. So even the future ones got named, numbered there. So. And now my parents have 30 grandkids, and I think that's it. So they don't know what I think my siblings are done having kids. But the first question to say is that's a lot of genetic experiments. So yes, it is. So that's part of what we'll be talking about today is what you can do with family history. And so I've done that on my own family. I've actually tested all my family's DNA, and everybody's related to me. And whether or not they're willing to admit that, it's a different question. But so this is what a DNA profile looks like. This is my mom, my dad, and me. And so the thing I can point out from this is that DNA shows I'm my parents' child. So not surprising, but that's so we actually have profiles framed on at home like this on the wall. So if you like to have family pictures or family profiles, right? So another view of my family, if you look at it from a genetic standpoint here. So this is how geneticists will list pedigrees. So you have circles are females and squares are males. And so my parents, me, and then my wife and 6 children. So I have 4 girls and 2 boys. And so 3 of them are in college right now and 3 are at home. And so I'll have the 3 that are in college will all be home next week. So excited to have all 6 of them back together. It'll be fun. So family history research is very much like a detective work, and you may recognize this address, 221B Baker Street. And a couple years ago I had a chance to go there, and here I am looking on Sherlock Holmes's pipe for his DNA. And so I gave a presentation the next day at the Royal Society in London, and I said I was looking for his DNA on his pipe, and someone came up afterwards and said, "Did you know that Sherlock Holmes isn't real?" And I turned right back to him and I said, "You do know I can't see DNA with a magnifying glass." So you have to have a little fun sometimes with science, and that's part of what it's all about. Coming together as families is important, and this is my dad kind of sharing some things from his family history, and I encourage you are we connecting cross generations now? And that's often I see people don't spend the time getting to know each other well enough across the generations now, and so we can do more, I think, to help in terms of sharing what we know with our children, grandchildren, and other relatives. So I highly encourage that. I mean, that's one of the things I want to give my kids for Christmas is a little bit more personal history stuff, so they can have that. Well, we live in a genomic age, and a genome is our DNA sequence. And if you weren't aware, that's the instruction book for a person or for any organism. And the structure of DNA was first described in 1953 by Watson and Crick. And so we still call the base pairing of the DNA strands coming together the Watson and Crick model for everything. The first genome was actually sequenced in Rockville. If you didn't know that, it's right here by Shady Grove Hospital. Right across the street is a place called the Genome, the Tiger, the Genome Institute, Research Institute, and they sequenced Haemophilus influenza there in 1995. So there's a lot of history here, and of course the Genome Project was done by the same person, Craig Venter, and then also by Francis Collins, who's right here in Bethesda. So they co-announced in June of 2000 the first draft of the human genome, and then the draft was polished off and finished on the April 25th, 2003 was the 50th anniversary of the discovery of the DNA, or the reporting of the double helix structure by Watson Crick when they published that April 25th, 1953. So in 50 years, we've gone from knowing what the structure of DNA was to being able to get a full sequence of the human genome. So it's pretty impressive in that regard. And since that time, there's been thousands of genomes that have been sequenced. So there's been a number of, over the years, since DNA testing has evolved over the years, and there's a number of techniques that have been used, historic methods for testing, starting really about 35 years ago in 1985 is really when DNA testing started taking off. And the restriction fragment link polymorphism used several different ways of doing that. You could do it looked like a barcode originally, and then there were single locus probes looking at just specific regions. I'm not going to go into all the details of this, but just getting down to the point that the technology has changed over the years. And what forensic testing uses today is this method here. This commercial kits have been available for the last 20 years, and uses fluorescently detected short tandem repeats. There's been a lot of advances with microarray chips, and that's what, if you get a 23andMe result or an Ancestry result, that's what you're getting. You're getting a microarray test for single nucleotide polymorphisms. Typically they run about 800,000 markers, and so you'll get about 800,000 letters, A, T, C's, or G's in a string that will be reported there. And now we even have more advanced parallel sequencing that allows full genomes to be sequenced, and that's what has enabled this. And the reason that's happened is the growth of, this is just a curve showing the cost. Originally the first genome cost several $billion, but then it's come down as more and more genomes have been sequenced to now it's down to the point where you said you paid $1,000 for a genome. So that's about, this was a couple years ago when the slide was made, and it was 1,500. So things have certainly come down. That's what's driven the growth and really the interest on the public, is having it within a cost range that you can afford. So for the science side, this is Francis Collins, who directs National Institutes of Health now, saying 11 years ago when he started the 1,000 Genomes Project, was his report. He says, in some ways, the application of new sequencing technologies is like building bigger telescopes. Just as astronomers can see further and more clearly into the universe with bigger telescopes, the results of the 1,000 Genomes Project will give greater resolution as we view our own genetic blueprint. We'll be able to see more things more clearly than before, and that will be important for understanding the genetic contributions to health and illness. So when they finished this particular phase of the project a couple years ago, they had done 2,504 complete human genomes at that point. Since that time, there's been well over 100,000 more genomes done, and it continues to grow. And so there's a lot happening in this area that's enabling more information to come out to connect with what does this mean from a clinical standpoint. And that's the question for how will this help medicine? Only time will tell. But certainly having more information can be beneficial. So what's fueling this growth? The new sequencing technologies has helped. Having more powerful computers so you can crunch all of this data and store the information and share the information. Really the globalization of science, meaning that now you can get samples from all over the world and connect and share information between things that are happening in the UK or in China or elsewhere. The interest of the public in ancestry testing, I think, has been a big driver. And now there's millions of people that have gotten their DNA tested. You can see a big portion of the room here have received ancestry testing in some way. There's been value to genetic research, so there's medical research, but still a lot to do there. And then 11 years ago, there was a passage of a law called the GINA, the Genetic Information Non-Discrimination Act. And the purpose of that was to say if someone's DNA is sequenced and is known then, that people can't be, health insurance companies can't discriminate against them based on the DNA that they have. So that was the purpose of that law. And I think that's helped fuel the interest in public of ancestry testing. So there was actually a push to, in 2007, Francis Collins held a meeting here in Bethesda along this very lines. He wanted to understand how much information do you need to know about identifiability? How well can you identify somebody? What's the minimum amount of information to identify somebody? And so this was published in Science, and the efforts to say, and see here, genomic data are unique to the individual and must be managed with care to maintain public trust, right? And that's really a key thing. Is there's always a proper balance between managing and encouraging genomic research and protecting privacy and confidentiality of research participants will not easily be achieved, right? We can't do it easily because that information is becoming more and more accessible, and with the sharing of information with the internet. So this was pointed out 12 years ago, and this conference was held to help encourage the passage of the GINA Act, which was passed the following year. So I participated in this with about 30 other people, and it was interesting to be part of the conversation. We were discussing the privacy balance and it's been interesting to see now with the Golden State Killer case and other things that have come out, is the impact this has had on the public understanding of what you can do with DNA information. So as we get into genealogy discussions, we'll come back to the forensic stuff a little bit later. But as we talk about what can you do in terms of family history testing, this is a slide that I got from a genetic genealogist in Charlottesville, Virginia. And she points out here that what you can do with DNA, you can confirm a genetic relationship. If you want to know that someone is a son or a cousin, a first cousin or something, that's possible to do with DNA testing. We can confirm and supplement a paper trail. So you may have, well, I'm a Butler, I want to know if someone else is in the Butler line, if that connects, you can do that. You can give hints about other names, other family or surnames to investigate if you're trying to research a particular branch. You can reunite with lost relatives. There's been certainly a number of stories where people have been able to connect with other relatives because of DNA testing now. And it can put you in touch with new relatives you didn't know. And that might be from anonymous donations to a sperm bank, and suddenly you have there's been movies made about this, lots of brothers or half-brothers or sisters that you didn't know about before. And so you make these connections because of DNA. What DNA cannot do, it cannot prove a lack of relationship. You can't fill in all your family tree with DNA. It can't be a substitute for your traditional research. So you're still going to have to do genealogy. Genetic genealogy will be helpful, but it will not answer all of your questions. It cannot give you an accurate percentage of all of your ethnicities from all your ancestors. And this is really important because I get this question all the time. I got a test done and it says I'm 22% Swedish and my brother is 50% Swedish. Are we really siblings? No, it's like, yes, that certainly is possible. And the last point here is this will not solve all your mysteries. And so speaking to this group, I want to help realize that, right, that you can't solve all your mysteries with DNA. So there's a number of companies that do DNA testing that have grown. This is a couple years ago I made this slide. There's a few new companies that have gotten involved, but FamilyTree DNA was really the first one to get into the business back in 2000, and they're located in Houston, Texas. Oxford Ancestors is in Oxford, England. And you may have heard of a book, The 7 Daughters of Eve. Has anybody heard of that book by Brian Sykes? So he's the guy who started Oxford Ancestors and has written several textbooks or popular books to try to help people find interest in what you can do with DNA testing. Sorensen Genomics and Relative Genetics have been purchased by Ancestry a few years ago. And so Ancestry is really the biggest player now in the field. And then 23andMe would be the second biggest player. So they have really grown, and there's MyHeritage as well, has over 1 million samples that they've tested. So the last numbers I heard, they were up at around 15 million, and they were like 7 or 8 million, but I think that keeps changing. But there's a rapidly growing interest from the public. The last numbers I've seen that have been published were in February, and there was 26 million people that had some kind of testing done with these companies. So this is from a couple years ago. There was a report in Nature about kind of the growth here, and you see the business growth opportunities that were being seen. So we're now in 2019, and they were estimating at that point there would be about $175 million business in this area, and they expect to be up to close to $350 million by 2022. So you can see the rapid growth that is projected because of the interest in this. So why are people doing this testing? This is a cartoon that FamilyTree DNA had a couple years ago. You don't look anything like that long-haired, skinny kid I married 25 years ago. I need a DNA sample to make sure it's still you. And so that's a skeptic, right, in terms of looking at their spouse. And so that's done with just a swab inside of the mouth, those who have done that particular testing. Now, if you want to learn more about genetic genealogy, I highly recommend going to this website. This is the International Society of Genetic Genealogy, ISOG, and they have excellent resources for all the different companies. It links comparing the prices, different types of tests that are provided, and things that are available in terms of what's out there. There's also a journal that hasn't published much recently. This is from a couple years ago. But the Journal of Genetic Genealogy, and the editor for that was local here, was from Olney, here in Montgomery County, and so Whit Athy, I assume you know Whit, was the editor of this journal, and so he is not doing this anymore, I believe, and so the journal hasn't flourished as well recently. But there are a number of great articles on there. This is one we wrote in 2008 at their request. So how do we address the nomenclature of how the particular markers are being named so that the laboratories producing the results can share them to the customers in a consistent way? So the problem that was happening back in the mid-2000s was that a sample would be sent to multiple laboratories, to FamilyTree DNA and to Oxford Ancestors, for example, and they would come back with different results from the same person. And people were like, what's going on here? How come I'm getting a 10 here and 11 over here? Well, it's because of how they describe the results. And I'll illustrate that in just a second. So what's important to keep in mind as we talk about these things is that the DNA testing itself can be the same between a forensic test, a paternity test, and a genetic genealogy test, but how it's used and the conclusions that can be drawn from it can be different. And so we'll talk about some of the things that can come from that. So what we're doing with genetic genealogy is a direct consumer. You as a consumer is sending a sample to 23andMe or Ancestry, and you're then getting a sample result back, and then you're doing something with that and trying to make a comparison. So you can only compare the markers that have actually been tested. So if you test the markers that are done for forensic testing, you can compare to other samples run with those same markers. However, if you do a genealogy test with different markers, you can only compare that to other markers that have been run, the other samples run with the same markers. So the other thing to realize when we look at this is an eye chart for you here that looks at the amount of sharing on average across autosomal DNA tests among close relatives. So what we're doing in forensic testing is we're just looking against yourself. So if you're the one that contributed a sample, like a blood stain at a crime scene, then the person that would be the person that contributed that, you're comparing DNA directly back to that individual. So the question sample from the crime scene is being compared to the known sample from the individual, a direct comparison. When you're doing paternity testing, you're then comparing just a small group of people here. That might be the father and the mother against the child. And so you know the child, you know the mother often, and then you're trying to figure out the alleged father. Is the alleged father the biological father? And so that's the testing that's being done. If you're doing genealogy testing, you may want to know all of these different things. The relationships between, is someone a third cousin, or is someone an aunt or uncle, or someone a grandparent? And so it's a much different question you're asking, and there's different implications that can have from a genetic standpoint with that. So what's being shown here as well is you have different percentages. So if you look on this chart, if you're comparing yourself to yourself, 100% of the time it's going to match up, right? If you're comparing yourself to your father or to your mother, on average 50% of your DNA will come from mom and 50% will come from dad. If you're comparing to your grandfather or grandmother, 25% of your DNA will come. If you're going to great-grandparents, you're down now to 12.5%. So you see at each generation you're cutting in half the amount of information that's being connected and shared. So when you start looking down some of these lines out to cousins, we get out here to a third cousin, and less than 1%, 0.78% on average, is going to be the same for a third cousin. So you can establish a connection in many cases, but it's not going to be as strong as having a closer a connection to a parent or grandparent. Does that make sense? So as you get further away, it's going to have more of a dilution effect. So the other thing to keep in mind is different other types of testing. This is mitochondrial DNA. And what mitochondrial DNA testing will do is allow you to look, and I'll explain a little bit more of the genetics of this in a few minutes, but you're connecting across maternal relatives. So a mother will share with her children her mitochondrial DNA and so on. So these individuals on the maternal side, maternal great-great-grandmother, great-grandmother, grandmother, mother, and the individual, the self here, would have the same mitochondrial DNA unless there's a mutation that occurs. And so on average, it should be the same. The same with Y chromosome testing will give you that portion of the pedigree that you're looking for. So if you're trying to figure out, do I have a paternal relative, grandfather, great-grandfather, and so on, you can make that connection with the Y chromosome test. So this becomes important. What's your question? And when you're trying to ask and to address a specific connection you're trying to make genealogically.
Relatively speaking, how many base pairs or markers are in the Y DNA and mitochondrial DNA versus the whole genome?
There's much less. So the mitochondrial DNA is 16,569 bases, or 68, depending on how it's counted. And then that can vary by a couple bases, but it's around 16,500. So that's fairly small relative to 3 billion, right? The Y chromosome, we're actually only testing usually a few markers along the Y chromosome, sometimes 12 up to maybe 100 or so. And so by looking at those 12 or 100 markers, you can then get a pretty good idea of what's going on with that portion of the Y chromosome. The Y chromosome is about 50 megabases in size. So it's, again, fairly small relative to 3 billion on total. So when you're doing genetic genealogy, you really have to consider these three points here. You have what genetic variation are we looking at, what types of markers, and how are we going to measure that? And is there, from being at NIST, we worry about how measurements are done and the accuracy of those measurements and stuff. So that's certainly, I spent a lot of time thinking about the first one. The second one is where the genetics comes in, and this is where we're looking at the inheritance. What I just illustrated, the complicating factors. Where are you on this particular chart if you're trying to connect to a cousin? And the fact that you have less than 1% on average that's going to be shared with a third cousin, that means you're not going to be able to answer that question as well. It's going to be more challenging to do that. And then finally, you need to frame your question, and that's where talking to somebody that's done these types of tests before can be very helpful, because what's the question you're trying to answer? What types of samples should you compare? Now, it's fine to send off samples for yourself and see what you hit on the database, but if you want to know a specific, am I related to this specific individual, and invite them to also have testing done, you need to know, what are you trying to answer? What's the best genetic test to do to answer that question? Is it a mitochondrial test or a Y chromosome test? Is it an autosomal marker test, which is what's offered by the big companies now? So again, emphasize this, DNA results will not answer all of your questions. So what's being measured? The primary thing that was measured early on was short tandem repeats. And I'll explain and illustrate what those are in just a second. These cover all the chromosomes, the autosomal chromosomes, or chromosomes 1 to 22. And then we have the sex chromosomes, X and Y. And the Y chromosome testing, for surname testing, can give you 12 markers, 37, 67, or 111 YSTRs. That's from FamilyTree DNA that offers those particular tests. And then the X chromosome test can be done, in some cases, it's typically done in grandparent testing or half-sibling tests because of the specific genetics of what you can answer with an X chromosome. The markers that are being used most commonly now are the autosomal SNP markers that are being tested by FamilyTree, by Ancestry, and by 23andMe. And they're getting back a letter. So you're getting a string of letters that would be located again on markers on chromosome 1 to 22. And they typically test more than 500,000 markers currently. And then finally, you can get a full sequence. And this is a string of letters. And so, has anybody had their mitochondrial DNA tested? Okay, just a couple of you. So that, what you get back from there is, if you get a section of it, sequence, you may get 400 bases, 400 nucleotides. Or if you get the entire genome done, you'll get 16,568 or 69 or whatever you have. That's the entire mitochondrial genome. So that can be very useful in some situations, but again, that's your maternal line. It's not going to follow, usually in Western societies, we trace our surnames by the paternal line, by the Y chromosome. And so the Y chromosome tests are typically more common for surname uses. Okay, so this is one of the problems that existed early on with this, was the nomenclatures. So this is a section of DNA that is found in one of the tests that are being performed that we had just in our paper that we published a few years ago. And you can see that the sequence here can be named differently. So if we look at this top one here, if we call the sequence, this is all the same sequence. If we call it AGAT, there are 6 of those. If we call it GATA, just move it over by 1 letter, now there's only 5 of those. And then there's not another A out here, so that sequence finishes there. If you move it over 1 more base and call it an ATAG, notice there's only 5 of those. And then if we moved it down 1 more letter, we'd go TAGA. And so that's all describing the same data, but 3 of them call it a 5, and only 1 will call it a 6. And so what we encourage in this particular article is, let's have a standard way to describe those results so that everybody can talk to each other and be able to communicate. So we encourage everybody to push the sequence, the start of the sequence, as far as possible to the left, just like reading. And so therefore we would call it AGAT as the repeat, and that would be 6. So the number that would be reported here would be a 6, and that would be put into the database for searching. So when you do a search, everybody with a 6 would come up on that search, like, instead of a 5 or a 6. So that's just illustrated here, and versus a 6 versus a 5. Okay, so we can test the Y chromosome, or we can test mitochondrial DNA. And so again, the Y chromosome, you're passing on from the father to the son, and that's without change. so the entire Y chromosome is being passed on, unless you have a mutation that can occur, which does happen usually about 1 in every 1,000 generations or generational events that will occur. And we can use these short tandem repeats, STRs, or we can use SNP typing to give a bigger picture view. So where are these within a cell, an illustration of this within a cell? The nucleus of the cell, that's illustrated here, is like the computer control center of the cell. And within there's a single copy of each of the chromosomes that you're inheriting from your parents. So you'll have 2 copies of each gene that you're looking at from autosomal markers from the nucleus, one coming from mom and one coming from dad. There's only one nucleus in a cell. The mitochondria, on the other hand, there are hundreds of copies of mitochondria per cell. And therefore, you have lots of copies of mitochondrial DNA. And this is why, when I worked at the FBI, this is why we would look at mitochondrial DNA in hair. We could take a little sliver of hair and digest the DNA out of that, and mitochondrial DNA is found in hair because there's lots and lots of copies. And so you can sequence that section of mitochondrial DNA, make a connection between that hair and the person it came from. The problem, though, is that the siblings and any maternal relatives will also have that same mitochondrial DNA. It's not unique to the individual. But mitochondrial DNA is found in the outside the nucleus of the cell, and it's the powerhouse of the cell, and so it needs more energy, and so you're only getting that, though, from mom. And the reason that is, we'll illustrate in just a second. So this is an illustration of the 3 billion bases. Several have asked about the 3 billion bases, and that's coming from what's in the nucleus of the cell. And we have the autosomes, which are the 22 pairs. Again, 1 copy from mom, 1 copy from dad. And then we have the X and Y, or XX, for the sex chromosomes. And then we have the mitochondrial DNA, hundreds of copies per cell in the cell cytoplasm. And so the reason that the mother is donating the mitochondrial DNA is when there's a fertilization of an egg by a sperm, the sperm is contributing just the nuclear DNA. And the sperm's mitochondria are not, don't survive. There have been a few rare cases where that's been demonstrated, but that's extremely rare. So typically the mother's mitochondrial DNA is what will be passed down to the child because the mother's egg forms the basis of the zygote and then the formation of everything. So again, just an illustration of that. So dad is the one that determines by contributing an X chromosome or a Y chromosome whether the child will be a male or female. And the mother is contributing the mitochondrial DNA and an X chromosome. So the current scientific thinking that most of our DNA, 99.9 or so, is going to be the same, but that still means that 0.1% still means there can be 6 million differences between people on average. Okay, so yeah, so 0.1% would be 6 million out of 6 billion, because you have one set of 3 billion and the other set of 3 billion, so you have 6 billion bases. So if you look again on a pedigree chart, at each generation you're passing on half your DNA basically. So we don't look at it from this way. You might look at it from a genealogical standpoint. From a genetic standpoint, we flip that pedigree, and as I mentioned before, we have males as squares, females as circles. The parents are the horizontal lines. The offspring would be the vertical lines, and then if they're deceased, we would have it that way. So this would be my pedigree. My great-grandparents are deceased. My wife's great-grandparents are deceased. My father-in-law is deceased here. My mother-in-law is still alive, my wife, and then our children. And so that enables you to kind of quickly, a geneticist can look at this, or a genetic counselor can look at this and figure out information about the genetics of someone. All right, so of course you have siblings you can test and other siblings there. But on average, your autosomal DNA you're going to get 1/8 of your great-grandparent DNA in that way. So, but the Y chromosome, these are the blues there show the Y chromosome that would be in common. My Y chromosome would be the same as my father's and grandfather's and my two sons' mitochondrial DNA. On the mitochondrial side, then, my kids all have mitochondrial DNA from my wife and through her mom and her mom. So if we review this, we have Anakin, Padme, and then we have Luke and Leia. And he turns into Darth Vader, and he says, "Luke, I am your father." And he says, "I need a Y chromosome test to verify that." So that certainly could be done if Luke Skywalker wanted to do that, and we'll find out more about the rest of the Skywalker lineage, I guess, next next Friday. So, so what's involved in doing an ancestry test? You pay a company money. Okay? Very important here. So as the double helix or the dollar bills turn there, right? So you supply a sample of your DNA by swabbing the inside of your mouth and collect the cheek cells. Then you wait several weeks for the company to send you your results. You try and understand your results. That's the key thing. You search the company's database of DNA hoping to find a relative. You try and recruit other relatives to contribute DNA so your results will be meaningful there. And then you set up a website and you blog about your success or failure, right? And of course, recruiting more relatives takes you back up to the top. The company gets more money, and you keep going down to that. So that's I think it's important to keep that in perspective. There are things you can do with ancestry tests. Will not solve all your questions. So this is the type of information that gets returned. If you go to Ancestry, this is 23andMe, and you'll get this, and you get to meet this nice lady here who says she's 47%. I love that, 47.1%. They have that level of precision.
The illusion of accuracy.
Yes, the illusion of accuracy, excellent. So, and then so a certain amount of Chinese and everything there. Well, what's interesting is this same lady, if you go back, this was, I made this slide a couple years ago, I went back and looked, it changed. Now our Jacqueline here, who's shown on the page, is now 50.1% European, right? And now they break it down to British Isles and French and German and all this Scandinavian, 0.2% Scandinavian, and then they break it out, the Vietnamese. So what's happening? Why is this number changing? And that's because the data sets that they're comparing it against have changed. And that's what's important to realize, that this is where, in some cases, I don't want to use the word hocus pocus maybe, but it's to the level of don't rely a whole lot on those percentages, okay? They don't tell you a lot. And so if you go and look, so for example, if you look on Ancestry's website, this is what they say. How could my estimate change over time? Your ethnicity estimate is based on the data we have and the methods we use to compare your results to that data. Because we're always collecting more data, and our methods are constantly improving, your estimate may change over time. How can my DNA change? Don't worry, your DNA hasn't changed. Good, they got that right? Right. What has changed is how much we know about DNA, the amount of data you have available, and the ways you can look at it for clues to your past. You are still you. That's very reassuring that that's been done. So this is, I think, an honest blog report. You know, DNA can't find all your relatives. And there's a number of complicating factors. One, we have recombination that's occurring. Whenever you have every single generation, you're getting a reshuffling of your DNA. And that introduces ambiguity. And this is the point Jim brings up, that he thinks in some cases you can go out further. You may be able to do that in some specific cases, but on average you can't, because the further you go out, the shuffling is happening each time. Unless you have a very unique card that's going to be carried on, that's going to last through multiple generations. You have a random chance of inheritance of each, like if you're drawing from a deck of cards, that's going to, which allele goes, or which characteristic from which parent goes to an individual is entirely random. And I'll illustrate that in just a second. Over time, though, the original signal that's part of that family tree can be lost, that portion of the tree. Even though you really have that ancestor in the past, you may not see them in your own DNA or in a testing of a relative that way. And then finally, you have mutations that can occur. So this is the third complicating factor, that over time in inheritance, sometimes these numbers will slip, like a 15 will move to a 14, or a 15 may move to a 16, if you're doing a link-based measurement, an STR. Or you could have a SNP typing. So the SNP mutations occur much less than the STRs, which is why they're used more now in ancestry testing. So, and again, lineage markers are not impacted by the first two, recombination and random chance of inheritance, but they are impacted by the last one, which is mutation. Okay, so let me illustrate this with a pedigree here. So this is a marker that's found on the chromosome 19. It would not normally be tested for genealogy, but it's being tested for forensic testing. And so I had access to a 3-generation family here, and so I was able to test, and I color-coded these. So the question becomes, which one, who's getting grandpa's DNA of the grandchildren, and who's getting grandma's DNA? And so you can see this really nicely by the way I have it color-coded here. So if we look at the first one, we have a son here, and the son is a 15-16. So the 15 comes from mom, the 16 comes from dad. Okay? Now they have a daughter who's a 13-15, and the 13's coming from dad, and the 15's coming from mom. Okay?
Because the 16 is the Y?
No, no, no. This is autosomal. So this is markers coming from chromosome 19, and right. And so we're just passing on. You're going to get one allele or one character. You're going to get the number from either mom or dad, and so you can see the 13 and 15 again coming same. Which notice it's the same. These two sisters have the same. And the brother. No, the brother here is a. Yeah, this is not a brother. This is a husband.
Oh, but he's also a 13, 15.
Correct. He's a 13-15.
Oh,
So sometimes the DNA is the same between
okay.
a spouse, and what you have then, if the spouse has the same DNA, the children will have 13-15. In this case, we have no clue, is that 13 or 15 coming from mom or dad? So this is where you have the ambiguity. Okay? So if you look at this across this chart here, we can tell that the first grandchild, you can see that 15 is being carried through because the mother here has no 15, she's giving the 14, and therefore the 15 has to come from dad, and that 15 is coming from grandma. So in 3 generations, the grandma is contributing. The grandma here is giving that 15, the 15, and we can't tell who's contributing at this point. So this is a nice illustration. Grandpa is still the grandpa, but we're not able to detect him at this level, but 2 generations later, with this particular marker. Now, if we test another marker, grandpa may show up there. Or another 100 markers, or another 100,000 markers, you may be able to test them. You may be able to detect them. So that's how this genealogy works there. So this is the type of illustration that, this is from Kitty Cooper, who does a lot of genetic genealogy. And this is just an illustration of her brother shares 2,810 centimorgans, and that's a measure. A centimorgan is roughly 1 million bases of DNA. And so what she says here is 52 segments with 175 centimorgans being the longest are able to be shared. So she has these color-coded by her brothers, her first cousin, second cousin, and so on across this. And so this is a mosaic of kind of the illustration of where inheritance is coming from for these types of things. And again, you're getting different percentages from different, as you get further out from your relatives and so on. So this would take more to explain. I'm not going to go into great detail here. So this is an article published in Science in 2007. At this point, this is now 12 and a half years ago or so, there was a big boom in terms of the growth. It's even grown more, but they were making some points just from a scientific standpoint. Commercially available genetic ancestry tests have significant scientific limitations, but there are serious matters for many test takers. And they point out in this article, these tests should not be taken as determining the race and ethnicity of a test taker. They cannot pinpoint the place of origin or social affiliation of even one ancestor with exact certainty. More recently, there's a group called Sense About Science, which I assume is probably similar to your group here, that they're in England, and they address all kinds of interesting topics. I don't know if you've read any of their articles before. They have some excellent articles on, senseaboutscience.org, and they have a whole thing on genetic ancestry testing. And so I'll point out a couple of the things that they illustrate here. The same history that you receive could equally be given to thousands of other people. And then I have another highlighted part there. For the most part, these tests cannot tell you the things they claim to. They're little more than genetic astrology. Now, what they're talking about there is the ancestry percentage. Okay, they're not talking about the ability to connect with a cousin. That's a different question. So, and they pointed this out here. Again, DNA cannot do it alone. To answer a specific question about individual ancestry with any degree of confidence requires a combination of historical records and genetic information. So you're making the point that genetic genealogy, the genetics part is great, but you also need the genealogy piece, right? The family history research. So my own experience, so I sent in a sample to Ancestry, Father's Day special, 89 bucks. It took me 12 days to get the results back. So that's why I spit it into a tube. And how useful, it provided an ethnicity estimate. Okay, so in the first results I got back, not very useful. But there is Ancestry functions. You can connect to second and third cousins, which has happened since then. This is what the box looks like if you haven't done it. This is the email you get. It says Woohoo, you got results. Your results are in. And that's what I first got back. 97% European ancestry, with most being from Great Britain and from Western Europe. And then there's trace regions, and you'll notice the less than 1% Irish, okay? And then my missing 3% in that study was some African, according to this, and some Native American. So the point, though, with this is that anything below 5% is in the noise, and just ignore. It's not. So these are the regions that were being tested at the time I did the test. So they had 105 regions in Africa, and they've just made groups of samples and declared this group is from Togo, or this group's from South Africa, or whatever. And that's kind of how they do these classifications. So my original estimates are there on the left, and then these are my new estimates. And the last time that I checked, so it's changed, and actually there's been one more change since then. I didn't make a new slide to illustrate that, but I'm changing. My DNA is changing. No, it's not. So, but you can see that the percentages are changing, and now notice I'm up to I'm up to 2% Irish now, right? And yet the Butler Line, I'll show you that in a second, is Irish. So right, so they got me up to 2% now instead of my 1%, less than 1%.
And they've got you in a particular country now.
Yeah, they're getting more specificity. So here's my. They make these nice fancy charts available to you, so you get this. This is science, right? So we're excited about this. And then when you start looking at what does this mean, you have to be very careful. Again, the smaller percentages. Here's my Irish link, and then I have some Togo link, which I have. There's no indication. My sister's a professional genealogist, so I have my genealogy done back 10 generations on all lines, pretty much done.
Nobody from Togo.
So nobody from Togo. No. So, but there's probably
Togo.
There's somebody in Togo that has a Butler DNA. Maybe the Butler DNA got loose or something and got it in Togo. So this is looking at family search. Just going back. My ancestors have been in the United States for over 300 years. In Ireland before then, and then so I definitely have an Irish Y chromosome, and then to France and England and France before that. So this is probably a similar line to many others would have in similar fashion. And actually there's a Butler Castle in Kilkenny, Ireland. And if you go to Ireland, this is one of the tourist attractions, and my 20th great-grandfather is the one who donated this. He's the one that built this castle in 1391. And then they lived there until 1935 and donated. They got tired of paying the property taxes. And so they donated it to the city, and now it's part of the tourist trap.
This is genealogical research, not genetic.
Correct. This is genealogical research. I'm making the point. I just showed you I'm less than 1% according to Ancestry, Irish, and yet the paper trail, and also all the other Y chromosome tests that have been done with other butlers from the Irish lines, I have the same Y chromosome. So it makes the point that you can verify something genealogically, but you can't get back 33 generations with DNA. No. So another experience, FamilyTree DNA, a little more expensive. Results, I got the Y chromosome test, the family finder, autosomal, and this is the Y chromosome. You get back a series of what the haplogroup is and other matches.
Ooh,
that matching with other butlers there. And so no surprise, my Y chromosome matches other butlers. I've tested all my father, my sons, my brothers, their sons. We all have the same Y chromosome that's being tested here. And then to point out, if you search in this is Y Search, the database a couple years ago. I searched this, and I found that there was only 17 out of 20 were butlers. So the butler did it. That's what this is from a forensic standpoint. However, if you search some of the forensic databases, you would find that there are no matches. And in this case, there's now several hundred thousand, but 39,000 samples that were tested at the time. So what does this mean from a forensic test? If you're comparing the Y chromosome, you may not be getting an accurate percentage of what really exists in the ancestry. So I have a colleague named Catherine Butler Gettings. So she grew up as a butler in Virginia, and her father has the exact same Y chromosome on the test that's commonly done in forensic testing as I do. We both have our genealogies back more than eight or nine generations. I know when my ancestors came to Virginia in the early 1700s. Her family came to Virginia about the same time, So the closest we can be is 8th cousins. We have the same Y chromosome. Not she doesn't, but her father does. So we're probably 6th or 7th, 8th, 9th cousins. Our 5th great-grandfathers differ at least. So that means there's potentially thousands of male butlers that live in the Virginia area that have my Y chromosome that could be committing crimes. And if that's what's all that's being tested, that would be a concern, right? But yet a search of a forensic Y chromosome test shows that there's no matches. And so it would give you a false sense. There was another test that was done a couple years ago on Thomas Jefferson with a Y chromosome test. Just to illustrate this point, they did not dig up Thomas Jefferson to do this, but they found 5 living relatives that are shown down here, and they tested their Y chromosomes now Thomas didn't have any sons that lived. He had an uncle though, Field Jefferson, who has those 5 lines they tested. They all have the same Y chromosome, so they assume that's the same Y chromosome that Thomas Jefferson would have. Then Eston Heming's, the question is Sally Heming's 6th child, Eston Heming's, is son of Thomas Jefferson. And so they found a direct relative of Eston Heming's, who was alive, and he had the same Y chromosome as the Jefferson line. Okay, so then the question becomes, and they tested someone else who claimed to be Thomas Jefferson's son, a descendant of him, and he does not match. Okay, so the question becomes, can ancestry testing verify that Thomas Jefferson was the father of Eston Heming's? So you could have had a false paternity later. So maybe Thomas Woodson really is Thomas Jefferson's son, but then there's a break in this line so that the person tested today that thinks they're the son or the great-grandson of Thomas Woodson is not. So does DNA prove that he's a father of the slave's children? No, it can't prove that. You can just say that it's a Jefferson, right? There's 25 other male Jeffersons that they're aware of that lived in Central Virginia at the time. His brother Randolph was known to be very friendly with the slaves, so that's certainly a possibility, but we cannot prove it. Now, the point is with genealogy and genetic genealogy, you can't prove some of these situations. I just use that as an illustration. So I'll finish with what's happening now. So the Golden State Killer case happened 40 years ago. An individual who was committing a lot of crimes all throughout California. The crime scene sample, a sexual assault kit, was tested just recently, about 2 years ago, and was examined for genetic genealogy markers. And then it was uploaded to a genetic genealogy database known as GEDmatch to locate potential distant relatives of the perpetrator. Again, not knowing, they had searched all the regular databases. They've been working for years on this case. I actually spoke at a conference in California with a bunch of prosecutors the month before this case broke. Actually, the prosecutor that was involved in this case spoke right after me at this conference. And they were desperately looking to try to make a connection. And so they used genealogy to do this. Family trees were created, and the detective then, detective work, hundreds and hundreds of hours of detective work, reduced the number of possibilities of suspects. Finally, they went and using information from the DNA, they were able to go and look for a specific individual. They collected a confirmation sample from the tissue from the trash and the door handle from when this individual parked at the mall. And then they were able to then identify this as being Joseph DeAngelo. This has been on the news a lot in the last year and a half since this happened. And he's awaiting trial right now in California, in Sacramento, for this connection. So what made this investigation possible? The first was that you had a genetic profile that could be tested. So there's a single source sample, not a mixture of DNA, but a sample that had enough quantity of DNA that could be tested. And so I consider this as like, with a fishing analogy, this would be like the bait to go fishing. You have a large number of people that are in genetic genealogy databases, people that have contributed to testing. So millions, at that time there was about 16 million that were in databases. There's more fish in the sea to go fishing for. We have a high density test, marker test, and that's basically a tighter fishing net with 500,000 SNPs. It also used an ancestry testing, so they could say there's probably likely an Italian individual that was involved and someone with blue eyes. And so they could use other records, like records from the DMV and so on, to be able to make that connection. the people sharing the results with the community, because we had GEDmatch, they could the law enforcement could discreetly search and go fishing there, just taking the time to go fishing. And finally, the confirmation to make sure you hook the right fish was done with the regular testing, the STR testing, and that's what will be presented in court. So I wrote a blog on this back on DNA Day earlier this year that you can go and read on the NIST website. It goes through and talks about this. So if you just search genetic genealogy and DNA and my name, you can find it. Yeah. That describes basically what was done in this particular case and the analogy I just gave with the fishing. So genetic genealogy databases now are being infected by the possibility of criminal investigations. And so the authors in this particular article was published in Science back a year and a half ago. The erosion of limits on crime-solving technology may well be inevitable, and it threatens our collective civil liberties and opens the door to socially and politically unacceptable genetic surveillance. There's a lot of concern with how will this be used. Just about 2 months ago, this is Tom Callahan from the FBI lab, published this on responsible genetic genealogy. The Department of Justice has issued a policy by how genetic genealogy should be done to try to protect people's privacy as much as possible, and that's being emphasized. But there's been changes to the database. So GEDmatch started in 2010 as a place where people could just upload their data and share it. So you could be tested in Ancestry and 23andMe and still compare your results. In April, after the Golden State Killer case, they immediately put an announcement on, said that law enforcement was doing this, and they effectively opted everybody in with the chance to opt out. If you want to get out of the database, you can do that. It's up to you. And so about 1 million people were still in the database, and that's what allowed a number of investigations to be done. Over the last year and a half, there's been, well, probably close to 100 investigations aided, and people, serial killers that they haven't been able to catch for years, have been caught with this. But in May of this year, GEDmatch decided to change the rules to opt in so that people would have to check a box to say, I want my data to be searchable by law enforcement. So it changes things. And suddenly, the database dropped down to 0, and it's growing. I think it's around 200,000 or so now that are in the database that's saying, I want to contribute to this. And then just last week, GEDmatch was purchased by Verigen, which is a company that does work in forensic space that works a lot with law enforcement. So there's a question, how will this data be used in the future? We don't know. That's something else. So things are changing there. So that's what I had, just in terms of presentation.